Welcome to Gradcast, the official podcast of the Society of Graduate Students at the University of Western Ontario.
Hello everybody and welcome to Gradcast Podcast. I'm your host, Alex Mozinski. I'm joined by co-host
Tyson Davis. Thanks for joining us.
And today we are joined by our guest, Anish Engineer. Hello. So the first awesome question is, your last name is Engineer.
Yes, it is. Yes.
I totally thought that was a typo at first.
I get that a lot. I feel like when people first meet me, the first question that they ask is, "Are you an engineer?" And my answer to that is no. I would never be an engineer because I hated physics. I don't know about you, but I could never understand physics. So that was from the start. That was just no for me. But yeah, that's my last name.
That is amazing. I loved physics. I almost went a physics route, but I loved biology more.
I almost became an engineer. So
I almost became an engineer too. There you go. The three of us all been engineers in our own way. Anyway, that is a really cool last name. Is there a sort of
I think it goes back, I'm of Indian descent, so I think back in the day, probably my grandfather's grandfather was an engineer by profession, and I think that's how the last name stuck. At the time, India was under the British rule, and I think they identified people based on their profession or what they did. So because that family was into engineering, that last name was given to them, and it stuck around for generations, I guess.
I guess that makes a lot of sense. Like, I know someone whose last name is Shoesmith, and they would have probably at some point in that family lineage been shoesmiths. Or actually, the first name Taylor literally means a tailor, like person who makes suits.
And the last name Smith is so common because it referred to blacksmiths during British rule, and they were the people that didn't go to war because they were the ones making the weapons.
Exactly.
So Smith prevailed as a last name over other last names.
I wonder if my last name's Polish for blacksmith. My family heritage is blacksmith, so Mozinski, guys, probably doesn't mean blacksmith, but maybe.
You never know.
Very cool.
Yeah, I know. I looked this up a few days ago, actually. My mother's maiden name is Wheeler, and it comes from Weller, because apparently somewhere back in the lineage they were well diggers.
Yeah.
Oh!
Yeah.
I mean, that's a cool way to identify people, but I just don't know if it would work in today's day and age, like if you're a scientist.
Alex Scientist. That'd be a cool last name. That'd be
Alex Brain Research.
Yeah. Alex Neuroscientist.
There you go.
Tyson Math.
Mathematician.
Math. There's a book series that I read called The Chronicles of Prydain by Lloyd Alexander when I was young. The High King's name was Math, so that would be kind of cool.
There you go.
Anyway, enough about names. Names are cool, but we're here to talk about a little bit about graduate student research. So, Anish, what do you do?
So I'm a master's student, a first-year master's student in the Schulich School of Medicine and Dentistry, specifically in the Department of Physiology and Pharmacology.
PhysPharm.
Yeah, PhysPharm. That's for short. Actually, a lot of people get PhysPharm confused with physics and pharmacology because it's the short phys. I'm sure it's a short form for physics, I guess.
Yeah.
So, but we're not physics. And sometimes I even heard one thing that pharmacology, someone addressed in mail a letter to the Department of Farm Ecology because they thought it was the same thing as pharmacology or ecology. There's a lot of mix-ups with my department, but
phonetically that makes sense, quite a lot of sense. All right, so what is kind of the focus of what you do? So you're in PhysPharm. That's a really awesome program. I like it a lot because it's very broad. You could be studying the kidney or skin cancer or really even very basic respiratory neurobiology if you wanted to in that program. So
the program's interesting because it goes all the way from basic science to what we like to call as bedside and bench to bedside science, so all the clinical science. So it's a good combination of the two, and sometimes they intermix. So specifically my research involves cardiovascular development. So cardiogenesis or in utero, how does the heart develop?
So heart development. Wow. So tell us a little bit more specifically about sort of your role in researching or learning about heart?
So I'm more concerned about the pathological side of heart development, and the pathologies that develop. So mainly congenital heart disease and how misstep in heart development can lead to defects in the heart, morphological defects, and how that can affect newborns and you later in life.
So by defects, are we talking like, a semi-leaky valve or something
or a hole in the
more
heart?
severe than that?
These defects range from like something minor, like a minor hole in the heart, to something major, like major outflow tract defects. So like the great vessels that come out of your heart, they could be going into the wrong chambers of the heart, and that could obviously cause you major problems once you're born.
Those are like inferior vena cava and superior vena cava.
And even like the pulmonary arteries and the aorta, they could all be transposed and be in different locations because of the complex nature of cardiac development. And if you're born with a defect like that, your chances of survival are very slim. So it's important to do research in this area just to figure out why these mishaps occur and how we can, prevent them from occurring, and potential therapies that are available.
That's really amazing. The heart, to me, is really fascinating. It was I think when I took like basic physiology, like an early year undergrad, like cardiac physiology was probably like tied just about with neuroscience. So I'm the guy who's like ties a lot, physics and biology, neuroscience, and cardiac physiology. So that's really cool. So how do you go about conducting your research? So you're looking at congenital heart defects. Is there one in particular that you're looking into? Is there, at what point in life are you looking? Like in utero, at the development there, or things happening in utero pathologically, and then ultimately are you looking at what would be the effects of this pathological change in an adult, and kind of what systems are you using to look at this?
So we study our congenital heart defects in mice. That's our animal model of choice. And my project is more centered around the effect of maternal diabetes on congenital heart defects. So specifically pregestational maternal diabetes. So when the mother is diabetic and is hyperglycemic and has no control over blood sugar, that has an effect on the growing child inside of her. And that can cause heart anomalies later in life, and at birth as well.
Okay. You and I are really like almost two peas in a pod, because right now I do ALS research, but I was having a tough time deciding on grad research as well, and one of the things I was going to look at was maternal malnutrition on brain development. So moms out there, if you are really careful about your diet, it could have profound effects on the outcome of babies to be. So be really careful.
Yeah, and I think that has a lot to say, especially nowadays in today's time. A lot of people are being diagnosed with diabetes earlier in life, especially women and young children are being diagnosed as diabetic. And if that goes undetected, and if they do have a child, that can cause problems, major problems, such as congenital heart disease.
Okay. So I guess before we go any further into the possibilities for developmental problems under diabetic conditions for moms, what would you say is the outcome if a person is diabetic? Maybe they're genetically predisposed to that. They get diagnosed with diabetes back in high school. In order to have the healthiest baby possible, what would be something that moms could do in order to do their best to ensure that happens? Because life has to go on. You can't just put it off because, oh my gosh, I have this terrible affliction. What do I do?
I think controlling your blood sugar would be the number one thing that has to be done, because it's seen that a high blood sugar causes something called reactive oxygen species to be very much present in not only your body but your developing fetus's body, or your developing baby's body, and that causes a lot of cellular mishaps and a lot of cellular problems. So the control of the blood sugar levels is most important, and that can be done through various just lifestyle, food intake, as well as drug therapy. Okay,
so keeping the blood sugar down. Or I guess normal. Not low.
No.
So how I guess how variable does that get? I'm definitely not up to par on my knowledge about that.
So the model that we're using right now in my lab is we diagnose diabetes anything above 11 micromolars of blood sugar, and that's what we use as a start. And throughout the pregnancy, these mouse mothers, their blood glucose goes very high and exceeds almost 30 micromolars, and that is almost a kind of like a representation of what would happen, in a real life situation if a mother hadn't controlled her diabetes. And throughout pregnancy, as we eat more, as a woman's body changes, that would mirror that effect.
Okay. So micromolars would just mean basically the amount of sugar in the blood.
Yes.
It's kind of like grams per liter. How much is in a particular unit of blood?
Yes.
Isn't a mole related to Avogadro's number?
It is.
It is. It's the number of particles per unit.
6.02 times 10 to the 23.
I like how you can really like math. So.
I can bring the math back.
Basically.
Good old Avogadro. Okay. So, getting back, I guess, to the effects on the developing heart now. Mm-hmm. So what specific abnormalities, I don't know if you're far along enough to have seen them yet, but what do you expect to see in what you're looking for?
So the heart is probably one of the most complex organs that we have in our body, and in fact it's the first fully functional organ to develop in utero. And what it is, it's a combination of many different pools of progenitor cells, or many different pools of stem cells, basically, that all come together in a harmonic, orchestra kind of, and all form different parts of the heart. So one part of the heart is from a different lineage than another part of the heart. Specifically, like the right ventricle is from a different lineage or heritage of cells than the left ventricle.
So wait, okay. You're blowing my mind here.
Yeah.
So different groups of cells actually come together to form the heart?
Yes.
It's not from like one cell lineage?
No.
In the developing brain, like there's all sorts of cool things that happen with folds and cell differentiation, and I always assumed that the heart was made up of heart cells.
Yeah.
Not a developmental biologist at all. So they migrate from different places and make the heart.
And that view, the old view that the heart used to come from a single cell lineage, used to be the view that even developmental biologists had for probably around 15 years ago. That kind of changed when a new discovery was made of a new pool of progenitor cells that no one had really known about before, and they found that this cell line really does make up a lot of a large portion of the heart. So it also shows how paradigm shifts occur in science. How we thought that, the heart is just made from one, cell lineage, but no, it's actually over a combination of three different, different types of cells that make the heart.
Wow. So is that like different types of muscle fiber cells? Because the heart's a muscle.
Mm-hmm. Yes.
But or is it the cells within the heart actually are doing very different things? And would these different cell types then be more susceptible to whatever pathology is?
Yeah, exactly. Yeah, so the actual muscle, the muscle of the heart comes from three different cell types, primitive cell types. And one of the cell types, it's called the second heart field, is the most susceptible to external forces, and that can lead to congenital heart defects. So that's what my lab is studying right now. The effects of diabetes, maternal diabetes, on a cell type called the second heart field.
Jeez.
Yeah.
So, wow. What, I guess, to kind of really drill back into this, so what kinds of pathologies, really specifically, would you see?
So congenital heart defects are probably they are the most common type of birth defect, of live birth defect. They affect 1 to 5% of live births. And of that 1 to 5% of live births, a ventral septal defect is the most common type of congenital heart defect. So, which is basically, as you have put it before, a hole in the heart. It's a hole between the two large chambers, or the two large ventricles in your heart, and that causes a mixing of blood flow. And you mix oxygen in the blood with deoxygenated blood, and that has implications not only when you're born, but later in life as well.
So it would have a really reduced efficiency, basically, of your cardiovascular
It might even go unnoticed at birth. But then later on in life, when you do more exercises or insert yourself, you can really feel the effect of that.
And what would life be like for a person living with this particular type of congenital heart defect? Like what would that be like for them? How would they be able to run a 10-kilometer race?
Yeah.
And if so, how difficult would that be?
A lot of times people discover that they have some kind of congenital heart defect because they decide to run a really long race and they can't, or they have heart trouble and they can feel it physically. And in those cases, surgery is usually done to repair that. It's good that now modern medicine allows us to do these kind of intricate procedures of repairing a hole in the middle of your heart.
So, just out of curiosity, because I am probably the least biological person in this podcast, can the hole in the heart, so you described it as it happens on the boundary between the two chambers.
Yes.
Can it happen externally as well?
Not
shoot blood into the body? I have no idea, right?
Yeah, no, because the heart is surrounded by another encasing called a pericardium that will prevent that from happening. So, no, you can't shoot blood out that way.
Okay.
All right. So other than ventral septal defects, what other kinds of congenital heart problems might there be?
In my research right now, I'm seeing a lot of atrial septal defects. So there's also a septum between the two atria of the heart, which are the upper chambers, and that's a very common defect. And usually, we're all born with an atrial septal defect. And at birth, actually at the time of birth, that wall closes. It's really cool. And sometimes that wall can close at birth, so in that case you would have a mixing of those blood, and again that's what we call a minor defect, not a major one.
So it's not as bad if I guess in and out chambers of the heart are mixing than if the two kind of more middle chambers are mixing.
For all of us in our fetal stage, the upper chamber is always open, and it closes at the time of birth.
So the mother having diabetes during gestation can affect the baby even after the baby's been born then?
Essentially, yeah. That problem or that defect can go unnoticed and then affect the individual later in life.
How about afterwards? So after birth has taken place and they're still breastfeeding, do you think the effects would be extrapolated there? Do you think the weaning phase would be affected as well?
Yeah, no, that's really interesting. I don't think that the hyperglycemic environment in the mother itself would affect the breast milk, and then that would thereby affect the baby. So, but definitely in vitro, in utero, it would definitely, the hyperglycemic environment affects the child. And it's actually really interesting. It's only in the first, almost the first, I guess, in mice gestation, it's only during the first 14 days of pregnancy that the diabetes in the mother is really important, because that's when the heart develops. Again, it's the first organ to develop in the body. So comparing a mouse gestation period to a human gestation period, what does that 14 days mean in terms of the human 9-month cycle? Yeah, so a mouse gestation is usually from 18 to 20 days. Okay. Whereas the human gestation is almost 40 weeks, 39, 40 weeks. So it's almost like second 2 trimesters.
So it's almost like every day is 2 weeks.
Yeah.
Hour.
Yeah.
Interesting.
And in terms of the cardiovascular system development, then, do you think there's other issues with blood vessels then, like veins, arteries, and the overall circulation? Definitely.
What diabetes causes is something called endothelial dysfunction, and that can mess up the vasculature of the body body as well. And not only the heart, but also the lungs and other organs can be affected by maternal diabetes.
Wow. Okay, that's intense stuff. So I can tell that you're very knowledgeable about not one but 2 disorders that you're doing research on. I mean, you're essentially doing research on both diabetes and congenital heart defects.
Yeah, and I think that's what the Phys Pharm program really aims at. It's really a combination of different aspects of human physiology and pharmacology, and putting them together and then, researching like that.
That's amazing. And you seem pretty passionate about it. So why did you decide to get into this particular branch of research? Not why Phys Pharm, but why cardiovascular and diabetes research? Why grad school?
I think it all I think it was all chosen in my 4th year study. I was able to take classes that I was really interested in, genuinely interested in, and I was able to see professors that I really admired, and I really liked their research. And I was really interested in what they were doing in their field of study. So I think that molded my decision to go to grad school and decision to choose the lab that I'm in right now.
So were there any, courses in particular that you took then in your 4th year that really made you say, this is what I want to do?
It was probably a cardiovascular pharmacology course that was taught by my PI right now that I took. So that influenced me a lot. But also a lot of my 4th year had to do with the heart and cardiovascular.
Very cool. I think that's a similar story with a lot of grad students.
Yeah.
I mean, in my 3rd and 4th years of undergrad, I took 5 or 6 courses from the same guy, and he was a geometer, and so now I do geometry. It's a person at that point in your life influences you, and you admire that person, and it kind of melds where you're going to go with the rest of your life.
Yeah. So I probably missed it at the beginning, but who's your supervisor then?
It's Dr. Feng.
Dr. Feng?
Yeah.
Okay, so anyone who's listening, looking into really interesting research, Dr. Feng, what's the first name?
King Peng.
King Peng Feng. Doing some really awesome research here. So what else, when you're not doing this research as a grad student, what else do you do? I guess you did your undergrad here as well, so you know London pretty well.
Yeah, I did my undergrad here. I'm really into, student politics. So I'm, as well as, just doing my master's I guess in undergrad I was a part of the USC, and I was very active on it. I was a counselor and part of different committees on the USC. Now I'm trying to carry that forward in grad school as well. So I joined, internally I joined committees on Phys Pharm, but I'm also now running for graduate student senator.
Oh, wow.
As a part of the Senate elections that are upcoming on February 10th and 11th. So I'll be running for a position of senator, yeah.
Oh, geez, heavily into politics. When you run for prime minister, increase the funding for science.
Exactly.
Please give us our money.
Keep us going.
And I've heard that sentiment a lot, actually, that funding for research is really going down, and it all has to do it all goes back to the government and, their interest in research.
Mm-hmm.
Well, I think that's about it for this interview. We'll have to wrap up, but thank you so much for coming on Gradcast.
Thank you.
It's been amazing to get to know you a little bit, and I hope we can hear more from you another time.
For sure, yeah.
And if you run, you've got my vote.
Thank you. Yeah,
definitely.
Yeah.
All right, thank you very much.
Thank you.
Thank you.
That's all for this week. If you want to send us some feedback, or if you want to come on the show yourself, email us at gradcastradio@gmail.com. Be sure to hook us up on social media. On Twitter, we're at GradcastRadio, and look up Gradcast Radio also on Facebook. If you want to subscribe to the podcast is located at gradcast.podbean.com, and it's on iTunes. And while you're there, why don't you leave us a review? It really helps us out. We'll see you guys next week.